today for the first TAGx event of the year. Before we get into today's very exciting talk by Megan, I just wanted to take a quick 5 minutes to introduce you all to our single cell product that Megan used to generate all of her really cool data. And that's our single cell immune profiling kit, which some of you actually might know as simply the 5' kit. The goal of the next few slides is really to highlight what analytes can be measured with single cell resolution using this kit, and ultimately show you how you can take a multi-omic approach to studying your key immunology questions. So before I dive straight into the capabilities of the immune profiling assay, let's take a few steps back and look at the Chromium single cell platform as a whole. As you can see, the Chromium platform consists of an instrument called a Chromium controller, which is shown here. It's about the size of a shoebox and fits on your lab bench. And then into this instrument, you feed these disposable microfluidic chips. And then onto those chips, you can run one of 4 main assay types. And of course, all of them are listed here as well. So regardless of your target analyte, the key output from all these assays is really the ability to characterize cellular heterogeneity. And I do also want to highlight that for all of these assays, we have ready to use robust workflows, including demonstrated protocols for various sample types and turnkey software tools. So if you're new to the world of single cell and want to apply it to your research, but feel somewhat intimidated about where to begin, then we have solutions across the entire spectrum of the workflow for every one of our assays. But let's move on and take a closer look at the output from our single cell immune profiling assay. So the core of this assay is detection of mRNA, where we're capturing whole transcriptome 5' digital gene expression from single cells. Because we're capturing the 5' edited transcripts, you can also expand the menu of analytes to include full-length paired VDJ sequences from T and B cells, meaning you can also examine the T and B cell repertoire diversity alongside whole transcriptome mRNA. The way this works is you perform a PCR enrichment step from the CDA molecules that were captured from single cells, and you pull out the VDJ sequences. So what this means is there's no computational wizardry here to do the pairing of the alpha and beta chains if you're looking at T cells, or heavy and light chains if you're looking at B cells. You're detecting exactly what the cell is making, so the pairing information is coming from the cell itself. You can also detect cell surface proteins in addition to transcriptome-wide gene expression and cell surface receptors, all from the exact same single cell. And this is achieved by having DNA barcoded antibodies, which bind the cell surface protein. You can think of this antibody labeling of proteins as being akin to sort of FACS, but of course in FACS you use a fluorophore barcoded antibody. Unlike FACS, in our approach you're not limited by spectral overlap of dyes, which means the number of cell surface proteins that can be multiplexed is essentially unlimited. However, of course, there are always practical limitations to just how many things you can measure. But we do see commercially available panels of over 100 antibodies specific for different cell surface proteins being used routinely, particularly in the context of our immune profiling solution. So what is super cool now, on top of all the information you can capture from the exact same single cell, you can also layer on antigen recognition. Just like the other feature barcoding assay, this works by applying an oligo barcode indirectly to your antigen interest. So that way, when the antigen binds the T cell or the B cell surface receptor, it remains bound, and this oligo that is present can now be picked up by the assay biochemistry and used to infer the molecular identity of the antigen species present. So really handy for those folks for interest who are doing, say, antibody discovery work. So from just one assay, you can get whole transcriptome digital gene expression, full-length paired VDJ sequences, cell surface protein, and antigen recognition, all from the exact same single cell. As you can imagine, the ability to simultaneously examine cellular heterogeneity of the immune system, T and B cell repertoire diversity, and antigen specificity, all at single cell resolution, really has a broad applicability across 7 key areas of immunology research. So if you have a key immunology question that requires single cell resolution to address, then please reach out to us and we can talk further about how our single cell immune profiling kit can really meet your needs. So to finish on, I want to highlight 2 key initiatives we have running at the moment at TAGx. The first is our experience event, which is held globally on February 22nd, but also again at a more Australia-friendly time of 2 p.m. on February 24th. At this virtual event, we'll highlight our latest developments in single cell, spatial, and in situ sequencing, and really give you a sneak peek of what's going to be available in 2022, including some new advancements to our single cell immune profiling product. So I definitely encourage you to try and attend and check out all the exciting announcements that we'll have at this event. And then the final initiative we're running here is what we call Meet the 10 Expert. Here we encourage you to reach out and meet with us to discuss your single cell needs. We found this particularly useful for those who are embarking on a single cell experiment but just want some guidance to set them on the right path to success. So, here's my email address. Just reach out directly if you want to schedule some time to talk. You can also find a link to this on the Millennium website. And with that, I hope you got a good insight into the capabilities of our single-cell immune profiling kit. And I'll say thank you once again for attending and hand the reins over to Megan for her exciting presentation today.
Cool, thanks so much, Jeff. So I'm just going to share my screen. All right, there we go. Hope everyone can see this. All right, so hi everyone, my name is Megan and I'm one of the postdocs. I'm working for Michelle Boyle here at QIMRB. Sorry, just hang on, I just want to see, I hope no one can see this bar, but yeah, I think that should be okay. Yeah, so I've just been using the single cell technology to help to characterize TFH heterogeneity for helping to improve immunity to malaria. So in the Boyle lab, we work with malaria, and malaria, according to the most recent World Malaria Report in 2021, it has estimated, there is still an estimated of 240 malaria cases worldwide, with roughly 600,000 deaths due to malaria. And these figures are actually a lot higher compared to the figures in 2019, which is largely attributed to COVID that also happened at the same time. So more importantly, in the report, WHO actually stated that the global milestone set by them for malaria to reach by 2020 was off track by 40%. And so one thing that they did mention was that in order for us to reach the 2030 goals, this would require new approaches, new tools, and as well as better implementation of the existing tools. So in order to contribute to this, in the Boyle lab, we're interested in improving our understanding of the mechanisms that underlie the development of long-lasting immunity to malaria. And this is primarily driven by antibodies. So antibodies are important not just for natural, but also vaccine-mediated immunity to malaria. However, the development of antibodies is a multi-layer regulation. So it starts off at the first stage over here at the level of APCs, where cells such as dendritic cells and also monocytes, they get activated when parasites invade the body. When they get activated, they then activate the adaptive immune response, where CD4 T cells then get activated. One subset of CD4 T cells, known as TFH cells, then drive and support B cell responses to then lead to antibody production. So in the lab, we've got various projects that are ongoing that, look into all the different layers of regulation. And in that, we've got also an aspect within each of these projects where we've used single cell technology as well. So I've just listed them over here. I won't go through each of them because of time. But if any of you are working on this cell type using single cell RNA sequencing, we would love to, chat and know you better. So for today, I'll just be focusing on my part, which is looking at the regulation of TFH cells in malaria using the 10x genomics platform on the 5' kit. So TFH cells, as I mentioned earlier, upon activation, they migrate towards the B cell follicle, where they then interact with B cells to drive the germinal center formation. So it is at this site where important processes such as immunoglobulin class switching, somatic hypermutation will occur, and this will, and what happens also is that B cells will then differentiate into memory B cells or long-lived plasma cells, as well as make antibodies. So in humans, TFH cells have been commonly studied in the circulation, and they commonly have been observed to exist in 3 different subsets known as TFH1, 2, and 17. And the way that people have conventionally categorized them is largely by looking at 2 surface markers, so CXCR3 and also CCR6. What various studies have then shown is that each of these subsets are slightly different from each other in terms of function, and therefore vary in their capacity to support B cell responses depending on the disease that you're looking at. So just to quickly illustrate this, this is some flow cytometry data showing you the cytokine profile for the different subsets just from healthy donors upon restimulation. So for example, interferon gamma over here is mainly produced by TFH1, while IL-4 is mainly produced by TFH2, and IL-17A is mainly produced by TFH17. If we look at more cytokines, we can also see that the levels can vary depending on the subset that you're looking at, simply illustrating that each of the subset are different from each other. And this is a slightly different way of just representing that data set. So, just showing how they each have a slightly different cytokine profile. Now, one thing that we noted in the lab was that if we were to incorporate more markers to differentiate TFH further, such as CCR4, we noted that the cytokine profile, even within each subset, can look again different from each other, depending on whether or not they express CCR4. So what this made us realize is that perhaps TFH cells are even more heterogeneous than we originally thought, and we believe that this should be taken into account when studying their responses to malaria. So what is known about the TFH subset regulation in malaria? Not too much. So, 2 years ago from the lab, we've got a study where we looked into the TFH subset regulation in a volunteer infection cohort. So what happened in this cohort is that, malaria naive adults come into the lab, they get inoculated with blood stage parasites, where they then get monitored for about a month for drug screening purposes. But what we also have access is their PBMCs isolated at day 0 before inoculation, day 8, which is the peak of infection, a week later at day 15, as well at the end of the month. What we found was that the different subsets had a slightly different kinetic of activation, with TFH2 cells being reaching its peak of activation on day 8, whereas TFH1 reaching its peak at day 15. What was more important was that when we correlated the activation with antibody responses measured at the end of the month, it was only the TFH2 cells that were positively associated with functional antibody responses. In a slightly different study, published also from the lab just earlier this year, we looked into the role of TFH1 cells in a different cohort. So we looked into children and adults from a malaria endemic region, and we found that TFH1 cells were the main ones that were activated in children with malaria. Now, this was consistent with an older study showing further that the preferential activation of TFH1 cells in children with malaria was instead associated with atypical memory B cell responses. So because of this, in our lab currently, we're really focused on trying to understand how we can target TFH cells more specifically. In particular, we're interested in boosting TFH2 subset more so than the other two for malaria. So in order to understand this, we firstly need to understand how are the subsets differentially regulated over time. So in general, there is not much that is known out there regarding the transcriptional regulation of how TFH cells differentiate into the different subsets anyway. We also don't know if the subsets stay stably as one over time or can they interconvert. And finally, not much is known regarding genes that are specific for TFH2 cells. And so we hope that we can identify gene targets to boost only TFH2, but not the other two. So we resorted to single-cell RNA sequencing to help answer some of this question. One of our first attempts was performed a few years ago using the 5' kit, but on the version 1 and 1.1 chemistry. So in this run, we isolated TFH cells from one patient from the volunteer infection study at the 4 different time points. We had isolated cells for both gene expression and also VDJ analysis, but also had some feature barcoding that we included on the day 8 time point. So just some quick summary from this run. The first thing that we've done was, just to look at the different clusters, and it was important to us that we can at least identify the 3 different subsets that should exist. So it was good that we can find a cluster belonging to TFH1, TFH17, as well as TFH2. But we also noted that there were other minor clusters, such as a regulatory cluster and also one with high type 1 interferon signature. But one thing that we noted in this exercise was that it was quite difficult to predict accurately a cluster belonging to TFH2. This is simply because there is not much known in the literature regarding positive markers that define this cluster specifically compared to the other two. There's one rare marker, GATA3, that we can find, but we needed to use a combination of markers that mark germinal center TFH cells, and scoring that, we saw that the blue cluster over here was the highest, and so we think that this should be the TFH2 cluster. But moving on from that, we wanted to firstly look at the distribution of the different cell types over time, and we can see over here in this graph there's not too much of a change in the proportion of cell types over time. But if we did a global differential gene expression analysis, we noted that the biggest transcriptomic changes were detected in the comparisons of day 8 versus the other time points. So this is consistent with day 8 being the peak of infection. And then what I wanted to then quickly do was to, see if there's any subset specific level of gene regulation. So I took all the genes that were upregulated at day 8 that are common to the 3 comparisons over here. And I then just plotted, the expression of these genes across the different clusters, and you can see that certainly there are certain genes that are more so upregulated in one cluster but not the other, and therefore again emphasizing that this heterogeneity that is present within TFH cells must be taken into account when studying their responses. So with this, we were encouraged to really expand the study further by including more donors, and so that's what we did. But before I go into that, because of what we've learned from that first run where, it was quite hard to really know that the cluster that we're defining as TFH2 is in the TFH2 cluster, we thought that it might actually be beneficial if we could create a transcriptional reference map for the different TFH subsets in humans. So that's what we did. We had a side project where we isolated the 3 different subsets using, where we sorted for these subsets using their conventional markers from 4 healthy donors. We put them through, once again, the 5' kit, but this time around we've got access to the version 2 chemistry. But we also just isolated cells again for the gene expression and also the VDJ analysis. So what we hoped from this was that we could create a transcriptional map for each of the different cluster, and then we can project this onto our bigger query map, which is looking at all TFH cells in malaria. So we think that, using this reference map, we can then use this to annotate the different clusters that are present on all TFH cells during malaria, and then map their dynamics over time. So in this second run, we've isolated TFH cells from 4 additional donors from the same study cohort, and we've also put that through the 5' kit on the version 2 chemistry, again isolating for gene expression and VDJ analysis. So one thing that we want to do after that was to basically perform some clonal analysis where we could start looking at some of the clones and seeing how they get distributed in the different subsets over time, whether they stay within one or do they move around. So for today, I'm just going to focus on the reference map because this current data set, we've finished processing them, but it's currently still waiting to be sequenced. So I'm just showing you some data from the reference map data set. So first thing first, this is some clustering and dimensionality reduction analysis. On this UMap, it does look quite different from what I initially thought. I thought that, the 3 subsets should just, cluster distinctly from each other, but clearly that's not the case. If we looked at this filing plots over here, showing you the some of the traditional markers that have been used to segregate the 3 different subsets, clearly we can see that they are different from each other. However, it is really only at a single cell resolution, where we can incorporate more dimensions as well to perform clustering that we can see such a pattern. So in here, we can still see that there is definitely a bunch of cells within each subset that sit separate from the other 2. So for example, this bunch of TFH1 cells, they don't cluster with the other 2. Or, this minor cluster of TFH17 that don't really overlap with the other 2, indicating that there are cells here that are, just clearly different from the other 2. But the majority of them have other features that, are at play, causing them to cluster in such a way that they overlap with one another. So this really illustrates to us that we're probably on the right track, in thinking that TFH cells are a lot more heterogeneous than thought, and we're just beginning to uncover more of that. So to understand this data a bit more, I perform unsupervised clustering. I have currently just stopped at 9 clusters that I think should be sufficient to understand this data set. And over here, I'm just showing you the distribution of the different cell types within each cluster. And you can see that most clusters are made up of a mixed bag of the 3 different cell types. However, there's certainly certain clusters that are more dominantly made up of 1 cell type. So this is quite clear over here for cluster 2, where it's mostly TFH1. Cluster 5 has more TFH17 cells than the other clusters. For TFH2, it's not as clear, but cluster 3 seems to be the one with the most TFH2 cells compared to the rest. So this is just a slightly different way of representing this data. Now then, moving on, I wanted to start annotating this map. So I just looked at marker genes for each different clusters. Some of them are a lot easier to annotate. So for example, cluster 2 over here, they're strongly upregulating TH1 genes. Cluster 5 over here, they've strongly upregulated TH17 genes, but there are also other clusters like this cluster over here that has, strong upregulation of heat shock protein, false and gene markers, whatever that means. Another cluster here with type 1 interferon signature. But coming back to TFH2, once again, it's still an exercise in progress for us because there is not much out there known as to what are the specific markers defining TFH2. We noted that if we look into cluster 3, which has the most TFH2 cell type, they do indeed have some strong upregulation of certain canonical TFH markers, such as LEF1 and TCF7. So we think that this should be it. However, this is still work in progress. So one thing that I did want to quickly mention is that this reference map currently is made up of 4 different donors, and we haven't been able to finish demultiplexing this map into the 4 different donors to check for any batch effect that could be due to that. So that's still work in progress currently, and after doing that, we'll just confirm some of this. So moving on to some VDJ analysis. So I filtered out for cells that express 1 TCR alpha and 1 TCR beta chain. And these are the number of cells that I've gotten at the end, which is roughly 40 to 60% of the number of cells that I had for gene expression analysis. If we look at the relative number of the clonotypes, most of them are of single clones with only a handful that have expanded. So the first thing that I did over here was to simply look at the distribution of these clones, either according to the sorted cell type identity or according to the single cell clusters that I've annotated. And I thought this brought about quite a few interesting observations. So for example, there's definitely a large group of clones over here that came from the TFH1 cell type that get distributed into cluster 2, which is also the TFH1 single cell cluster. However, there are other clones that are distributed to the other clusters. So for example, a small group of clones over here that get distributed to cluster 0, which also had clones from TFH2 and TFH17. So this would suggest that there are differences in terms of TCR clones, which are causing the different cells to get distributed to the different single cell clusters, which would be really interesting to look into further. And then this graph over here, I'm just showing you how the different clones are shared between single cell clusters. Most of them are unique within each cluster, with only a handful that are crossing between clusters. So once again, this is still, work in progress. I looked at the VDJ dataset last Friday, and we really need to demultiplex this into the different donors and then, look into this further. But I hope within this short talk, I was able to convince you that the single cell technology is a very useful technology to help to uncover heterogeneity that may not be fully understood before from an immune cell type, even if we're looking at a subset of immune cells such as the TFH cells. So these are some of the future work that we'll still need to do from this data set, and then to hopefully reach the final point where we can map the dynamics of each of these subsets during infection, investigate the plasticity of these cell types, and finally to identify gene targets that are specific for TFH2 for malaria. So with this, I'd just like to end my talk by thanking the different lab members from the Boyle lab for their contribution, as well as our collaborators. And last but not least, all the volunteers and their families for making this possible. Thank you so much. I'll stop sharing.
Thanks, Megan. So everyone, there will be a short poll that should pop up on your screen. Please take a few moments to fill that out. If you have any questions, please type it in the chat, or you could raise your hands and we can unmute you. Megan, I don't see any questions as of yet.
I've got a question, Karen, for Megan. The multiplexing that you did based on SNP, so what was your decision to load a particular, how many cells did you decide on, how many donors did you decide to pull together, and how did you come upon those decisions?
Yes, that's right. Yes, sorry, I forgot to highlight that. So I know that we can use the hashtagging technology for demultiplexing, but we've actually tried previously on one of the 3' run to try to, use SNPs, and that was pretty useful. So we're like, oh, let's try to do that again this time. So in terms of the ratio, like how we combine, we tried to just be equal. So after sorting the cells, we did a cell count and just tried to load as best as possible equal ratio of each donor, so that hopefully we're not skewing the data set. Obviously we don't know until we demultiplex it.
Great.
Yeah.
Oh, and I guess a follow-on question from that is, how difficult did you find it doing the demultiplexing, given that 10x doesn't officially support genetic demultiplexing? So therefore we don't have a pipeline for it. So how did you manage to implement that in conjunction with Cell Ranger, and how difficult was it?
Yeah, very good question. So I don't know if he's here, but another postdoc in the lab, he's actually doing the demultiplexing. So that's not my part in the analysis. We don't use any bit from the Cell Ranger. As I understood, I think he's got his own platform, but I think there are a few other either packages or some other platforms that's out there. I don't think they're widely used yet. I think one of his first attempts, like a few weeks ago, he was like, this is okay, but we want to be a bit more sure. So we're trying out a different package. So yeah, I can't fully comment on the difficulty on that bit. We think hopefully it should be doable. But yeah, I'm not the right person.
No problems.
All right, it doesn't look like we have any further questions. Once again, thank you, Megan and Jeff, for presenting today. This session has been recorded, so I will be sending everyone a recording link.